Hi. So it's now down to the last keynote at the conference, and it gives me great pleasure to introduce John Hardy to come and speak, or should I say Professor Sir John Hardy, or is it Sir Professor? Whatever.
Just Sir will do.
Just Sir will do, exactly. Well, you know, along with a bow, anyway. But no, in all seriousness, it's a huge pleasure to introduce him, for him to talk to us about the genomics of neurodegeneration, you know, when he's such a key person who's brought that field forward. Of course, his contributions in this area have been very well recognised, so there's obviously the knighthood, but there have been the Brain Prize and many other prizes to boot in the field. In addition, I think it's really worth kind of noting why the contributions have come in this way and why that recognition has come. And it's because of these key discoveries. Genetic discoveries, I think, have really made a difference to how neurodegeneration is perceived in terms of its solubility. And maybe solubility is a good word when you're talking about amyloid deposition. But you know, obviously there's been the identification of APP mutations, and from that, the amyloid cascade hypothesis. There's also been TREM2 and the identification of that and the role of the immune system in Alzheimer's disease, the identification of SNCA triplications and their role in Parkinson's disease. And really, a lot of this work is now coming to clinical fruition, and that's probably the most exciting component. So the amyloid hypothesis, obviously, has been tested in a sense through the generation of drugs to clear amyloid, and those are now showing efficacy, making that hypothesis more than a hypothesis now. So this is sort of a very exciting time for neurodegeneration, where the genetics are beginning to reach fruition. But, you know, before actually just ending here and saying, you know, leaving it to John, I just want to say that the contributions of some individuals to fields is more than just the discoveries and more than just actually the papers. What they are is also a contribution to a sort of family of researchers who've come after. So I'm one of those. I think there are others in this audience who have links to John, and as he puts it, you never stop working for me. So there's a positive and a negative in this. But yeah, but, you know, I never stopped working for you, but I stopped being paid by you. That's the problem, right? So those two things are disconnected. But anyway, so it's really, you know, his contributions go beyond just those discoveries. They have kind of burgeoned a sort of family of researchers who are excited by it, interested in neurodegeneration genetics, and all the follow-up studies that have to come with that. So I'll now leave it to John himself to do the talking. Thanks very much.
Oh, thanks, sweetheart. You're
the quest for medical treatments and cures isn't just about slides and microscopes and what can you do about the symptoms. It's about families, it's about individuals, it's about stories of people who suddenly begin to lose themselves in a way that's frightening for them and terribly painful for those who love them.
A lot of people would say that Alzheimer's disease research, to some extent, started with Carol's family.
All of them were between the ages of 54 and 58, and it was at that stage that our life changed, really.
We put an advert in 1985, 1986 in the Alzheimer's Society newsletter because we were interested in studying the genetics of Alzheimer's disease. And the very first letter I got was from Carol Jennings, and she had drawn her family tree, described her family tree.
The breakthrough itself came in 1991. We got a letter in January telling us they had identified a gene that they believed caused this Alzheimer's disease.
When we found the mutations in amyloid in Carol's family, we knew that that is where the disease started. It's been a dominant feature of all work on Alzheimer's disease. Carol's family has what's called autosomal dominant Alzheimer's disease, and that means that if your father or mother has the disease, you have a 50% chance of inheriting the gene, and if you get the gene, you're going to get the disease at a very similar age probably to your parent.
She's a shell of the woman she once was, but she's still my wife, and so she's still part of who I am. We may not be able to change the cards that life deals with us, but we can change the game we play with the cards we've got so that future generations don't have to carry the same pain.
I'll just say a few more words about Carol Jennings, who really was a remarkable woman. And actually her husband, as you saw, is a pretty remarkable man. And in fact, her son is also a pretty remarkable man. He's now, of course, at 50% risk. And he and his father are both honorary presidents of the Alzheimer's Society, and both go out fundraising for the Alzheimer's Society. And her son is now, who also doesn't know his genetic status, comes into Queen Square to be part of the trials. When we found the gene, Carol was quoted in the Times as saying she didn't think that the findings would help her, but she hoped that it would help her children's generation. And I have to say that when she said that, I thought maybe we should be able to sort this out in 5 or 6 years. And this is 1992. But in fact, Carol was far more realistic than we were. And what she did, she didn't want to, the video's got a slight error in it. She didn't work with other families with Alzheimer's disease. What she did was she didn't want to see others with Alzheimer's disease. She became the national volunteer organizer for families with frontotemporal dementia. So we still, in fact, have an annual meeting of frontotemporal dementia. Well, both researchers and sufferers. And she was the person who organized it. She was there organizing it, organizing the catering, the boring stuff around meetings. She did it, and she was there at the meeting. And of course, she knew what it was like to be in those families, so she was just a good person to have around. And well, she's now very, very ill. Well, of course, it's a tragedy like all of these families, but she was a remarkable woman. Anyway, I'll talk some more. So I'm going to talk about more than half of what I say is going to be about Alzheimer's disease, but I'm also going to talk about the Parkinson's disease and also tauopathies. Very quickly, here are the pathologies. This is the pathology of Alzheimer's disease. Top left, you can see a cross section of the brain down the midline. In the middle there is the expanded ventricle. Top right is a cross section where the two lines are. Bottom left, the bottom left is the hippocampus on that area. That's shrunken. That's the part of the brain centrally involved in making new memories. And of course, that fits with the earliest symptom of the disease, which is a problem with making new memories. And then at the bottom there is the histopathology. Bottom left are tangles in the cortex made up of the tau protein. Incidentally, great work from Shaw Sherez and Michel Gerder here in Cambridge have really worked out the structure of these over the last 5 years. Central middle is our plaques. Those are about 1/10 of the millimeter across, made up largely of the beta amyloid protein. And bottom right is the amyloid deposits in the blood vessels, which are also A beta. I'll talk about those in terms of the complications of therapy at the end. I'll just say that for now what I'll say is I started work on Alzheimer's disease in, well, you don't need to know necessarily, but about 1980. And I went to meetings in 1980. There will be 20 or 30 people at those meetings, because that's how many people were working on the disease at that time. And of the 20 or 30, there'd be 10 or 15 different hypotheses about what caused the disease. So it was really anarchic. People could say whatever they wanted about the causation of the disease. And in 1983, Jim Gusella reported Huntington's linkage to chromosome 4, largely using the Venezuela kindred. And this was a new technology. I was a pharmacologist. I'd never heard of this technology of using genetic linkage analysis and what were then called RFLPs. And I read his paper and I thought, wow, instead of just looking at the end of the disease, you can use genetics to find out how the disease started. And I decided to try and change my career goal because I could see that my view was, if you could find out how the disease started and you had this as how the disease ended, you'd have both the start and the end of the disease process, and you could put it into a rational sequence of events, and then you could perhaps intervene in that. So that's why I decided to study genetics. Now, the original, I'm credited on both, I'm credited and cursed. Both are unfair, really, for the amyloid hypothesis. In fact, I said there were many hypotheses. Glenner was the first person to actually really say the amyloid hypothesis. His paper is top left. I'll read the key sentence because it's very small. What he did was he looked at the amyloid sequence from a case of Alzheimer's disease, derived in fact from the blood vessels, not from the plaques, and got a peptide sequence, about 20 amino acids. And then he did the same thing from a case of Down syndrome. People with Down syndrome nearly always develop Alzheimer pathology by the time they're about 45, and got the same sequence. And he said, and I'm sorry, my eyesight's not so good. This is, when he got the same sequence, this is the first chemical evidence of a relationship between Down syndrome and Alzheimer's disease. It suggests that Down syndrome is a predictable model of Alzheimer's disease. Assuming the amyloid peptide is a human gene product, it also suggests the genetic defect in Alzheimer's disease is on chromosome 21. Now, implicit in that, of course, is he obviously thinks that having 3 copies of chromosome 21 leads to overproduction of amyloid, and therefore that is really indirectly the amyloid hypothesis, and he is completely right. The next year, Colin Masters, who was sequencing the amyloid plaque cause, derived almost exactly the same sequence from the amyloid plaque cause. His group eventually cloned the amyloid gene and located it on chromosome 21. So they really should have both the credit and the blame for the amyloid hypothesis. Anyway, to move briefly on to Parkinson's disease, this is the pathology of the major pathology of Parkinson's disease. Top right is a normal nigra at the top of the spinal cord. The cells in black there are black because they contain melanin, and they're the dopaminergic projections which go to the striatum. Top left is a Parkinson nigra, showing the faded nigra, and that it's faded because a lot of the cells have died. Bottom right is a picture of the pathology, which is of a Lewy body, the predominant protein component of which is alpha-synuclein. I'll actually show you an EM of a Lewy body later on. So it's the primary pathology then is the synuclein pathology. Tangle diseases, here the nomenclature is a bit complicated. There's a hereditary tangle disease, FTDP17, with tau mutations. Here, the most common sporadic tangle disease, progressive supranuclear palsy, and here, bottom right, you can see a tangle in PSP. The other tangle diseases are corticobasal degeneration, And when you hear about CTE or dementia pugilistica, that also is a tangle disease. So these are the three types of diseases I'm going to be talking about. Now, possibly the simplest message to leave this lecture with is that in all these diseases, simply overproducing the protein, the deposited protein, will give you disease. We showed, led by Andy Singleton, that synuclein gene duplications and triplications led to Parkinson's disease, and then blocking on his name, and my eyesight is just getting worse. Yes, Dominic Campion. Sorry, Dominic. Gene duplications, amyloid gene duplications caused Alzheimer's disease, and tau gene duplications caused tangle disease. Very simple finding, but actually I think it's a very important finding, because what it's telling you is all of these proteins are close to their, what I'll call their crystallization threshold. All of them are close to their crystallization threshold. Slightly overproduce them and you'll get deposition. Now, of course, there are many other mutations. We and others have found other synuclein mutations. We and others have found other tau mutations. Many of the other tau mutations specifically increase the amount of 4-repeat tau which is generated. And there are many other APP mutations, some of which decrease the solubility of the amyloid which is produced, some of which increase the amount of amyloid which is produced from the amyloid precursor protein. So within this small number, because it is a small number of families, everything is consistent. Overproducing the protein leads to deposition. I'm making these points from a genetic perspective. The paper on the bottom right really influenced my views on this, and that's from Michele Vendruscola here in Cambridge, and he made the point, and this is one of the several papers from their group, that all of these proteins that I've mentioned are highly expressed, and all of them are close to their, well, crystallization threshold. So I'm making the point from a genetic point of view, but he made it also from a protein chemistry point of view. This is the family we started with. This is Carol Jennings' family, where we found the linkage in we found amyloid mutations. Actually, I can remember the date, November the 20th, 1990. And this is the family tree. And I'm showing this so you understand linkage analysis, which is really simple. We're looking at the top half of chromosome 21 in Carol Jennings' family. All of the people in black there have inherited the entire chromosome segment. The people, the 9th and then the 11th person who is not affected, the 9th person has got the top bit of the chromosome and is not affected, and the 11th has got the bottom bit and is not affected. So the simplest explanation is that the gene mutation must be somewhere between those two regions. The APP, amyloid precursor gene, is between those two loci. That story seems very neat. The paper at the bottom is a kind of the dirt from behind the scenes on the how we got there. We and many other groups made many mistakes. It was much more difficult. This work, which I've just described here, took a team of 6 of us about 5 years. So this simple diagram, together with the mutation, represented in 1990 30 person years of work. Now I think a good postdoc could do it in 3 months. So that's because of the technology and the fact we have the human genome now. But anyway, the dirt behind it, if you're interested in the dirt behind, I used to watch a terrible TV series called Behind the Hollywood behind the scenes. And this paper is my contribution to that literature. So we found APP gene duplications. It was immediately clear our families have been collected by Martin Rosser and I. We had about 30 to 35 families at that time. APP gene mutations explained 3 or 4 of them, and so there was obviously another gene, and that other gene, well, in fact there were 2 other genes as it turned out, presenilin 1 and presenilin 2, cloned by Peter Hislop. When presenilin structure was determined, the structure is given in blue there. It's a 9 transmembrane domain. When it was cloned, we didn't know what its function was, but because of work by Bart de Strooper and Dennis Selko's group, we quite quickly learned that it was responsible for cleaving both APP and, as it happens, many other substrates in the transmembrane domain. So in yellow is APP going through. It would actually go up through the ceiling and down through the floor. That's a truncated part of it. And in turquoise on the presenilin are the aspartic acids, which contribute the active site of the enzyme gamma secretase, which cleaves about where that line of red mutations are. So that's where gamma secretase cleaves. There is a beautiful series of papers, which I don't have time to go into, but from Lucia Chavez Gutierrez, which details exactly how how presenilin gamma secretase acts, exactly how it cleaves. In 1998, we were looking for a gene which caused frontotemporal dementia, which is in the form that we were looking for it is the Tangle-Oney disease. We and Jerry Schellenberg's group found tau mutations caused FT, frontotemporal dementia, and that led us to be able to make mice with tangles. This work was done by Jada Lewis, and we crossed the mice with tangles with mice that we had made with amyloid mutations. So we now had mice with both amyloid and tau, and what these mice showed us was that amyloid drove tangle pathology. When we just had tau mutations, and you can look at the central 2 histograms here, if you just had tau mutations, you had almost no tangles in the cortex, but if you cross those mice with amyloid mice, you pushed tangles into the cortex. I'm going to come back to this data later on, but this is, if you like, the experimental evidence for the amyloid hypothesis. It clearly shows that amyloid drives tangle pathology. So that led us to draw this simple diagram. And I think it's worth looking. I sort of believe this diagram still, but let's look at what's wrong with it. There's many things which are wrong or misleading about it. The first is that when I drew it, I thought of this as being a biochemical pathway, rather similar to glycolysis. You know, APP metabolism, amyloid on the outside of cells, leading to tau dysfunction and tangle inside. But if you think about it, glycolysis, you alter glucose concentrations over a minute, and the rate of glycolysis in the cell immediately changes in seconds. So biochemical pathways operate in seconds. And we know now, there's been time courses of Alzheimer development, that this process takes 20 years about. So clearly, that's not a biochemical pathway in the same way that glycolysis is a biochemical pathway. The second problem with this diagram is that there's a line there going from which kind of is implicitly a neuronal membrane. So you've got amyloid going to the neuronal membrane, and then leading to tau dysfunction inside. So the only cell in here is a neuron. That's the only cell, and I think that also is misleading. And the other implicit thing here also is that one supposes that there is at that membrane surface a receptor for amyloid, and that the amyloid is binding to a receptor. And, you know, despite the fact that we drew this diagram in 1998, basically the question marks in this diagram still remain question marks. And eventually, if a question is never answered, perhaps it's not a good question, and I think that that is the case. So I'll come back to that later. Now, amyloid processing. If you look up amyloid processing anywhere, if you look it up, there's 1,000 people have drawn this diagram. I think it's on Wikipedia. This particular version isn't, but essentially the same thing. And so I'll go through the diagram, and then I'll say what I think is wrong with it. So the right-hand triptych is, I like the word triptych. That's one of my favourite words. It's it's a Catholic upbringing. Greek Orthodox churches full of triptychs. So what you've got there is beta secretase in blue, cutting at the north end of the amyloid path of the molecule, and then presenilin gamma secretase cutting at the bottom of gamma secretase and yielding amyloid. The left-hand triptych is alpha secretase, which is either ADAM10 or ADAM17, cutting partway up amyloid, and then presenilin gamma secretase yielding a fragment called P3. So you can think of the right-hand fragment being the bad way, and the left-hand being the good way in a kind of cartoon language. So this is how the metabolism is drawn. I think it's got one important problem with it, and that is anybody who's worked with amyloid knows how hydrophobic it is. It's a transmembrane domain. If you're working with amyloid in the lab, you have to keep it really, it's very difficult to work in plastic tubes because it sticks to the surface, to any lipophilic surface. So if it's being generated within a membrane, why would we expect it to float off in that way, straight into the extracellular space? I'm going to come back to that later on. So what I've been saying relates to early onset Alzheimer's disease families. There are about somewhere of the order of 100 such families in the UK. We wanted to find the risk genes for late onset Alzheimer's disease. We already knew about ApoE on chromosome 19, which is drawn there on the right, found by Alan Roses. We had spent years looking by candidate gene studies for other genes involved in late onset disease without success. And it was only with the advent of GWAS that we had began to have, well, really good success. And here is one of the GWAS. They're continually being updated. I think you'll have had GWAS presented. The chromosomes are along the bottom, and the line across is the p-value. You have to do a Bonferroni correction. All the genes above the line you're allowed to declare as Alzheimer risk genes. And so now in this diagram there's about 40 now, there's perhaps 70 or 80 have been declared. A list of genes is pretty boring, even to a geneticist, but really, I think the key paper came from Leslie Jones. It's the paper on the bottom left. She pointed out that instead of being obviously related to APP metabolism, that what they had in common was that they were involved usually in the immune system, and in the context of the brain, that meant largely, perhaps not exclusively, microglia, and she wrote cholesterol metabolism. In fact, I think it would have been more correct to say lipid metabolism. That was entirely a surprise. Notice in the title of this slide, I put obviously in italics, because I think that, as I'll say, I think they are involved, amyloid related. There are some genes which are directly APP metabolism rate related. On the right hand end is ADAM10 in red, and that is at one of the alpha secretase, and we have found missense mutations. So it does support directly the importance of APP metabolism, but in a much more strong way, it points at microglia and the immune system. I'm going to take a little diversion. But in the early 2000s, I thought, we found all of these genes for dominant dementias. Are there any genes for recessive dementias? And so what we decided to do was go to our colleagues in Turkey, Ebbe Lohman and Murat Emery, and look for consanguineous marriages, which had led to children, actually they adults, but led to children with dementia. And these 3 families have been identified by them. They all got disease in their 30s. And what Rita Guerrero did was use homozygosity and exome sequencing to find mutations in the gene TREM2. I'll talk about the mutations in a second. I'll just say that the paper has this really clumsy title. And the reason it has this clumsy title is because this gene, these types of families have been previously identified by Lena Peltonen, who actually tragically was the director here at the Sanger Center until she died early of a rare cancer about 10 years ago. And she had identified TREM2 mutations, but her families did not have any bone disease. And so our clinicians had not realized it was the same syndrome. So the mutations that we found in these were, I'm going to read them, Q33, XT66M, Y, God, I have to get glasses, Y something C. Sorry about that. Now, what do you do when you find mutations? These are homozygous mutations. What you do is you immediately look in your own database. At Queen Square, we're very lucky. We've got about, because of Anita Harding, Nick Wood, and Henry Holden, we've got a database now of something like 40,000 exomes of people with neurological disease and their sequences. So you look in your own databases, and then you look now also, of course, on the databases that are available publicly. Have these mutations ever shown up before? Here so that's what Rita did, and she looked in all the databases, and sure enough, when she looked in Alzheimer's disease, these mutations showed up. This mutation once in a case, not in a control. These two, 3 and 2 times, 3 times in cases, 2 times in cases, and neither in controls. So 6 times in cases, not in controls. That on its own was nearly genome-wide significant. But this mutation, this variant, I should say, R47H, was present 22 times in cases and 5 times in controls. So this suggests that this mutation, when homozygous, gives rise to this rare disease, which we now call Nasu-Hakola disease, when heterozygous predisposes to Alzheimer's disease. I should say that I'm pretty sure that R47H, when homozygous, does not give rise to Alzheimer's disease. We've never seen R47H as homozygote. So this is our paper top left. The paper top right is from Decode, which published back to back with us. They found the R47H essentially by doing linkage analysis in the Icelandic population. we published our papers, and then I got a nice email from Tom Bird, who wrote the paper at the bottom, published in 1983. He had sent his family to Lena Peltonen, and she'd found TREM2 mutations. This predates that gene finding exercise, but he had pointed out that this weird disease, now called Nasu-Hackler disease, had some family members with Alzheimer's disease. So he wrote to me very politely and said, if you'd written this 30 years earlier, you would have beaten me to publication. So, and that of course is accurate, and in fact that's a very interesting family, very well documented by Tom. Now, at the same time as we were doing this work, we were also, a completely different group of people in the lab, were doing gene expression studies. What they were doing was looking at what genes responded to amyloid deposition in mice. What genes respond to amyloid deposition in mice? And in fact, the gene which responds the most is TREM2. So TREM2 is not only a genetic predisposer for Alzheimer's disease, but it's also the most amyloid responsive genome-wide. So we decided to look at this systematically, and this was work done by Mar Maturin and Dervis Saleh. They looked at gene expression, and at the top left there you've got a network of genes which go up with when you get amyloid deposition, and TREM2 is the gene which is most upregulated. All the others are upregulated slightly less than TREM2, but the interesting thing which Mar noted was that many other GWAS hits were part of that network. And so what that allowed us to say is there is a network of amyloid responsive genes in and genetic variability in many of them is associated with disease. So it's saying not particularly about TREM2, what it's saying is how well you respond to amyloid deposition is important. So Dervis decided to follow this up and use this as in a systematic fashion and ask these three simple questions. Is there an overexpression? Clearly there is, 10 to the minus 41. If we take out the genes we know about, is it still significant? So which other genes contribute? His paper is there at the bottom. And this is that network again with TREM2 at the middle, now expanded with more sequencing behind it. All the genes in red ovals are genes we already knew about. Genes underlined in blue are genes which independently showed association with Alzheimer's disease. So there's a network of genes which are associated with disease. I'll just mention one of the genes on this network, OAS1. OAS1 is we're working on this at the moment. OAS1 is an interferon responsive gene, and exactly the same haplotype is now known to be associated with bad COVID. So it's there's an interesting correlation, if you like, between bad COVID and bad response to amyloid deposition, which we're trying to follow up. So we followed this up as well now and looked at exactly when TREM2 upregulation occurs. And this really nice paper from Francis Edwards looked at uses the nanostring technology to look at when in the mouse brain does gene expression change. And what she has done is done this experiment in TREM2 knockout mice and in control mice. And what she sees is a series of genes, some of which TREM2 itself is upregulated only when the microglia come into contact with the plaque, not before. In other words, it's a specific contact. When the microglia touch the plaque, that's when they get upregulated. There's a bunch of other genes which are upregulated when that contact occurs. There are other genes whose expression can go up distal from the plaque, and what one suspects is that their upregulation is driven indirectly by, for example, cytokines secreted from the microglia which touch the plaque. So their upregulation is secondary. There's very few genes, there are some very few genes on neuronal or astroglial. So we've gone here from thinking that everything was going to be neuronal to realizing that a huge proportion of risk is microglial response to amyloid deposition. In fact, I've been trying to encourage Dervis to write a review called Alzheimer's disease is a disease of microglial exhaustion, because that, in a way, is what one starts to think. Now, how do we put this together? And what I've told you so far, I hope, is fact. This is, well, not fiction, but I hope that this is my idea of what's going on. And it's driven by reading these papers. ABCA7 is one of the Alzheimer risk genes, and that is a phospholipid transporter, and TREM2 is a phospholipid receptor. So when you hear phospholipid, what you immediately think of is membranes. And so this is what I'd like to suggest. Here we go, this is how amyloid metabolism was drawn. What I'd like to say is I think that amyloid starts like this. It starts building up in the membrane, and it builds up in the membrane and starts to disrupt the membrane. And this disrupted membrane is then what attracts the microglia. And if you get it's when this system fails that you switch from being amyloid positive to starting the neurodegenerative process. That's what my view is. Now, what's downstream of amyloid deposition? I had to say that, well, we started doing GWAS in path-confirmed Alzheimer's disease for reasons that I detail in a review with Valentina Escott-Price. I think the clinical GWAS for Alzheimer's disease are very imperfect because the diagnostic accuracy rate is only about 70%. And a diagnostic accuracy rate of about 70% is not good enough when you start to find, for example, genes involved in other dementias in what's labeled as an Alzheimer GWAS. So we started to do a path-confirmed GWAS. Those are necessarily very small, typically now we're about 2,000 path-confirmed cases. I had thought, though, that we could do a GWAS of all the individuals who'd had amyloid imaging. Surely their diagnosis was right, and I thought that we could add those to the path-confirmed GWAS. That turned out to be wrong, because what we actually found when we did a GWAS of PET-scanned amyloid-positive individuals, what we found was only ApoE and no hint of all the microglial GWAS. So that led us to think that we divided the process into two stages. Amyloid deposition, almost entirely ApoE dependent. Dementia in that context, microglia added on to the previous ApoE. So that, I think we've divided the process, and that notion is supported by this paper. I showed you our paper very early on in this talk, showing that amyloid drove tangle formation. That experiment was repeated by this group, led by Hansen. And he did it, though, in the context of a TREM2 knockout, and showed that in the context of a TREM2 knockout, in other words, paralyzed microglia, you drove tangle formation far more greatly. In other words, this is putting the microglial response between amyloid deposition and tangle formation. What I've said so far is consistent with work from the Randy Bateman group looking in living individuals with sporadic Alzheimer's disease, saying their major problem is not in amyloid formation, but rather in amyloid clearance. So I won't read this because this is summarizing what I've been saying so far. Moving on very quickly now, I promise you, yeah, I really promise you, to Parkinson's disease. Here's a Lewy body. I think of a Lewy body as a failed lysosome. I mean, that's my kind of simple-minded view of it. When you look there, what do you see? You see damaged mitochondria in the Lewy body, and you also see loads and loads of curled membranes. Synuclein is a membrane-associated protein, and so this explains the pathology, and it also points to mitochondria and, well, in this case, membrane-associated proteins in the disease pathogenesis. I'm going to be very fast here. These are the known Mendelian genes for Parkinson's disease. What do we see? Mitophagy, as we see mitophagy and the endosome-lysosome pathway. The GWAS, which was in the last but one talk. What do we see? A lot of lysosome and mitophagy genes. How can we draw this with the pathology? Well, I think these papers from Dimitri Krank allow us to bring these two things together. Dimitri Krank showed that if you overexpress synuclein, it inhibits lysosome flux, and if you inhibit lysosome flux, then you lead to a buildup of synuclein. So basically, putting together, in the same way as I put amyloid and microglia, this paper puts together synuclein and lysosomes. This is what I've said for Parkinson's disease. Doing the GWAS for tangle diseases is less informative still, and it's less informative because there's less cases, but what we're already seeing in the GWAS for the tangle diseases is elements of the ubiquitin proteasome pathway, and our work on this is led by Hugh Morris. And sure enough, tau is cleared largely, at least in part, by the ubiquitin proteasome, as this work from Karen Duff makes clear. So overall, we're having a simple idea. Early onset disease, Mendelian disease, often overproduction of the protein. Late onset disease, sporadic disease, largely problems in clearance of the same protein. I should say that this rather takes away the special idea about these proteins. Maybe there's nothing special intrinsic to these proteins. Maybe the thing that's special to them is that they are the highest expressed proteins of their disposal class. Now, why are there nearly always co-pathologies? So when you get a brain with somebody diagnosed with Alzheimer's disease, let's say at the age of 80, the pathologist will say the primary diagnosis is Alzheimer's disease, but there's also, for example, lots of Lewy bodies. So why is that? And we molecular biologists have been ignoring this idea of co-pathologies. We don't like it. We like simple pathways. Why is it we get co-pathology? Well, I think we get co-pathology because it isn't as simple as this. When you have protein disposal groups, it isn't like this. It isn't A beta microglia, tau ubiquitin proteasome, synuclein lysosome. The clearance systems is rather like the motorways going around the ring roads going around London. If you get a blockage on the North Circular, immediately you'll start to get a blockage on the M25, not because those roads are connected, but because you get spillover between the different disposal classes. And that's why I think that you start to get co-pathologies. So I no longer think of amyloid driving tau in the way that diagram at the beginning did, but rather I think of amyloid crossing over and blocking up a nearly failing system, as the London road systems are already nearly failing, and pushing it over the edge. And that's the connection between the pathologies rather than a direct toxicity. Now I'm going to move on very briefly, 3 minutes 53 seconds, onto the clearance of amyloid. I should say the first paper to do clearance was this one in 1999, immunization. It's interesting to know that why they did this story. Why did they immunize amyloid mice with? Why did they immunize with amyloid mice with plaques? They did it not actually to clear plaques, but because previously there had been work suggesting that if you pumped up the immune system, you might get tangles as well. They did it to make the pathology worse. There was papers from Pat McGeer and Joe Rogers suggesting that the immune system might be involved in tangle formation after plaque deposition. So they did it for exactly the opposite reason from the one that's advertised in the paper. That's just a dirty little secret. Now, we've had 20 years of amyloid, immunization not working. Why has it suddenly worked? And the answer is because, in fact, most of the previous antibodies were given to mice which already had plaques. Most of the original and most of the earlier papers, the antibodies have been given to mice before plaque formation. In other words, they prevented plaque deposition. They did not remove plaques. And these latest papers are the first to remove plaques. And that's the difference. These are the aducanumab. You might remember the controversy, one positive trial, one failed trial, and yet it was approved. I'll come to that. Lecanemab last year was shown to be efficacious and unambiguously. I think everybody accepts it's efficacious. This is the data. I'll start with the left-hand top slide. That is showing amyloid removal there. So it's a very effective amyloid removing antibody. The very top graph is the clinical response. And you can see that there is there's a cleared response. It's about a 25 to 30% slowing. It's a slowing, not a stopping, but that's the response. How do we explain this? And actually, I'll just give a shout out to this review from Eric Karen and Bart de Strooper, who forecast what drugs would work and what would not. And their forecast is this diagram here. What they said was you had to, and the left-hand side of the diagram is this, you had to remove amyloid from the brain, and after that removal, you had to run the trial long enough for the red line, the clinical efficacy, to become evident. So they're saying that trials have to remove amyloid quite effectively, and after that, you might start to get efficacy. This was a prediction, and these are the actual data drawn from the trials. And you can see donanemab there achieves this. Lecanemab achieves it. Let's start with lecanemab. Donanemab, what you're looking at there is the phase 2 results. The phase 3 results were mentioned in the press release about a month ago, but have not been published. So this is the phase 2 results, which would have predicted efficacy. And then you can see the two aducanumab trials. The lower aducanumab line is the trial which showed clinical efficacy, and the upper aducanumab line is the trial which did not show efficacy. So consistent with the hypothesis. And then the trial which has just failed, gantenerumab, clearly you can see based on this hypothesis, why it failed. It did not, it removed amyloid, but not effectively. So we now have then a way of removing amyloid, and it's giving a 25 to 30% efficacy with people in early disease. Can we get better at diagnosing earlier, and would we get a better result if we did diagnose earlier? I think we would, but that experiment has still been done. We can use genetics. This is our polygenic analysis showing that we can predict who's at high risk of disease, and Henrik Zetterberg and Amanda Hesselgrave are running biomarker analyses, and biomarkers show real promise, blood biomarkers show real promise at getting the diagnosis earlier. And so I think we can get better at diagnosing early. I don't think amyloid is going to be the be-all and end-all of Alzheimer's disease. We've still got a 25 to 30% slowing, not a stopping. It's very good to see this paper come out 2 weeks ago from Kath Mummery, showing that the tau antisense may show some efficacy. I've seen data showing, in fact, that the tau antisense not only reduces tau in body fluids, but it also seems to remove tangles, or to some extent. So really, I think we are heading towards an era of treatment of disease, which of course is why we do this. I think it's worth saying now that we are in, these are, I think, I would say these are the first 2 swallows of spring. We've got 2 drugs which work, 2 drugs which are very expensive, 2 drugs which are difficult to give because there's side effects which perhaps we can talk about, and will require reorganization here in the UK of our healthcare system if we're able for us to give them effectively. So, you know, it is the beginning of the end, I think, for Alzheimer's disease. I think we will be able to deal with Alzheimer's disease in rather a similar way to the way we currently deal with heart disease. But there's a lot of work still to do. I think there's other targets still to hit, and there's an enormous amount of political work and organizational work to do to give these drugs to the people who need it. Thanks very much.
John, thanks so much for that overview and really taking it from sort of a single personal story of a family all the way through to actual treatment. I mean, it's quite an achievement to get it that far, and a lot of people's work. Are there any questions? I know that we haven't got very much time, but maybe if we give us 5 minutes and then we'll go through to closing remarks. So, oh, thanks. Sorry.
Thank you very much, Doctor. Excuse me, I have 2 questions. The first one about the GWAS, you mentioned that it's maybe not reliable for some to give some results. Which method or technology do you suggest better than a GWAS in such cases? The second question is about the recent clinical trials to remove or to get rid of the amyloid. Which pathway can these drugs to remove? I mean, there's intervention with its formation pathway or destruction. That are my 2 questions, and thank you very much for this opportunity. Thank you.
So I think the problem with the GWAS not being reliable is not the technology, but the samples which go in there. I think we need to do GWAS on path-confirmed cases, and that's a difficult answer because there's so few path-confirmed cases available. So there's no short answer. When you do genome sequencing and you look for rare variants, it's a bit less of a problem in rare variants, because if you, let's say, find a rare variant in a few cases, hopefully you can go back to the clinical data in those few cases and find out whether they genuinely did have disease. So sequencing, looking for rare variants, is not quite as susceptible to the problems of GWAS in terms of misdiagnosis rate, because you should be able to go back, hopefully, and find out the clinical data. In terms of other drugs, I think that now people might go back to the drug categories they'd given up on before. And here I'm thinking of BACE inhibitors. We might think about BACE inhibitors were abandoned a while ago because they did not lead to clinical efficacy. One could think about selective BACE inhibitors as an adjunct to amyloid removal. One could think about that. And whether that'll be, whether the drug companies will have the stomach for that, I don't know. The original BACE inhibitors were not clean. They also inhibited BACE2, and I think that also was a problem. So it is possible that one could go to that sort of combination. Of course, what I also think is that we'll be, I hope, going to this type of trial where we do both an anti-tau drug and an anti-amyloid drug and see if we get an additive effect. But I think the key thing next is to give the same drugs that we have now earlier in the disease process. That, in my view, is the key, possibly easier goal next.
I think we've got one question online and then one question in the audience, and then we'll go to closing remarks, just because I think it's heading to close on time.
Yeah, so we have one. What about the link between the short-chain fatty acids produced by the colonic microbiome, which influence greatly microglial activity? Is the link between microbiome and predisposition to Alzheimer strong in sporadic case where genetic testing finds nothing?
I mean, I'm afraid my answer is brutally short. And that is, I don't know. I mean, I really, we've not looked at that at all. And yeah, we've not looked at it at all. If you'd asked me 5 years ago, I'd have said that's a daft question. I'm less, as you get older, you get a bit more humble. So I'm a bit less confident in saying that's a daft question anymore. It's worth looking at, and it is being looked at, of course.
I think there was one more question from the audience, from Sonia.
Thanks, John. That was amazing. I really like the way you synthesised that onset was at protein deposition or aggregation, and then the host response and the failure of clearance mechanisms leads also to disease. So I wondered whether you thought progression is related at all to the rate at which those clearance mechanisms fail, and whether there's any genetic basis for that idea.
That is a very, very good question, and one we're trying to investigate. The answer so far is we don't have any evidence. We've looked, and we are looking. We're looking in drug industry data to see rate of decline, and to see if we can do a GWAS of rate of decline. And the answer so far is that nothing. It's a very small study still. And we have one locus, but it isn't a locus we've seen before. So the answer so far, we don't have much data. That's basically where we're up to. It would be a lovely idea, but no data yet.